Yeah, okay.
This assignment, it involves the usage of Bpsp and Bpmp, where a specific sequence of nucleotides are given in order to determine its own protein. I will explain about Bpsp 54 first. What are we going to do? To analyze protein sequence using three tools or software, which are BLAST, InterProscan, and ClustalX. Firstly, compare the protein sequence against a non-reductant protein sequence database by using BLAST or FASTA. From using BLAST, the conserved domain are obtained. Then, we will further analyze protein sequence using InterProscan. InterProscan is secondary database that contain information on protein families, domains, and functional site. When such searches return significant matches or hit, this result help in get information regarding specific site, superfamilies, and also its alignment. Moreover, upon using the software InterProscan, the domain site is known and there is explanation for each domain. The first one is HAMP linker domain. It presents in histidine kinases, adenine cyclase, methyl accepting proteins, and phosphatases. Is an approximately 50 amino acid alpha helical region. It is found in bacterial sensor and chemotaxis proteins and eukaryotic histidine kinases. The bacterial proteins are usually integral membrane proteins and part of a two-component signal transduction pathway. One or several copies of the HMP domain can be found in association with other domains such as histidine kinase domain, the bacterial chemotaxis sensory transducer domain, the PAS repeat, the ESC domain, the GODF domain, the protein phosphatase 2C-like domain, the guanylate cyclase domain, or the response regulatory domain. It has been suggested that the HMP domain possesses a role of regulating the phosphorylation or methylation of homodimer receptor by transmitting the conformational changes in pre-plasmic ligand binding domains to cytoplasmic signaling kinase and methyl acceptor. GO terms. In biological process, this domain involves in signal transduction. In molecular function, it involves in signal transducer activity. This domain has integral component of membrane. By using ClustalX, the MSN, DND, and ALN information can be obtained. Later, PDB will be used again in BLAST in order to get a conserved domain. Then, using PDB ID obtained, and later it can be looked upon in PDB in order to know the protein name and also its secondary and tertiary structure. The PDB ID detected is 3ZX6, which is ID for HMP mutant.
I miss you. Across the
Next, I will proceed with Bpmp. The sequence obtained is number 131. The steps are similar to Bpsp. Firstly, the BLAST shows the conserved domain compared to non-reductant site, and later InterProscan is used to know the domain site and the family. Here the protein families was obtained using software InterProscan. The first one is peptidase M20. This group of proteins contains the metallopeptidase and non-peptidase homologs that belong to the MEROPS peptidase family. The peptidase of these clans have two catalytic zinc ion at the active sites bound by histidine, aspartate, glutamine, aspartate, glutamine, and histidine. The catalyzed reaction involves the release of N-terminal amino acid, usually neutral hydrophobic, from a polypeptide. The peptidase family has 4 subfamilies. First one, M20A, type example glutamate carboxy peptidase from Pseudomonas. Second, M20B, type example peptidase T from E. coli. Third, M20C, type example X-cis dipeptidase from E. coli. Fourth, M20D, type example carboxy peptidase from Sulfoloholus sulfataricus. Then second one, protein families, which is acetyl-ornithine or acetylysine deacetylase, also known as DAPE, this DAC. This clade of mainly archaeal and related bacterial species contain 2 characterized enzymes, and this acetylase which specific for both N-acetylene ornithine and N-acetylysine from Thormos, which is found within lysine biosynthesis, and a fusion protein with acetyl-glutamate kinase. From Lactobacillus, it is possible that all of the sequences within this clade have dual specificity, or that a mix of specificities have evolved with this clade. MSADND is obtained too, and from the BLAST PDB conserved domain are obtained, and the PDB ID detected are 2F7V, which is the ID for acetylcitrulline deacetylase complex with 1 CEO. It is
Okay.
나 혹시 몰라 경고하는데
잘 들어.
지금 위험해
So dangerous.
자꾸 나를 자극하지 마